My name is Bode-Dalby. I'm from the University of Bremen, and currently I'm working in the traffic project, which is mostly concerning traffic transfer efficiency in the Benguela current. And there I'm the postdoc in the sub-project 3 concerning the role of zooplankton for traffic transfer efficiency in the Benguela current coastal upwelling system, where we specifically compare the northern to the southern subsystem. Well, I chose to talk a little bit today about one part of my research, which concerns the role of zooplankton in the biological carbon pump. And yeah, I would like to go. Okay. So what is the biological carbon pump? Without it, the atmospheric pressure of CO2 would be around 200 parts per million higher than it is currently estimated. So it is, or it plays a huge role within the global carbon cycle. It starts with the phytoplankton incorporating the CO2 into organic matter, and it is made of these 3 major processes, which are the advection processes, the sinking of particles, which is also referred to the passive fluxes, and the third one is the vertical migration, which are also referred to as the active fluxes of carbon. So the phytoplankton organic carbon can be transported down from the surface to the deeper layers via physical transport as dissolved organic matter, or also by the formation of aggregation, while the largest part of phytoplankton is consumed by microzooplankton in the surface layers, while this in turn is then consumed by mesozooplankton, and also a fraction of the mesozooplankton is directly consuming this phytoplankton. So there are very different processes going on. The major one of the sinking particles is the repackaging of the mesozooplankton of phytoplankton or other organic matter materials into these dense, fast-sinking fecal pellets, which then enhance the sinking of particles. But the mesozooplankton can also attenuate carbon fluxes. So there are different processes going on, but zooplankton plays a key role in the biological carbon pump, mediating these carbon fluxes. And also they are doing large vertical migrations on a seasonal or on a diel scale. So these diel vertical migrations are also very huge migration on a global scale, whereas the general pattern is that during the night, the zooplankton moves up into the surface layers, then feed in these surface layers, and then during the day, they migrate down to the deeper layers, where they metabolize the organic matter that they've taken up in the surface layer during the night. So this is then actively transported down into this ocean interior. Just to have some numbers here, the zooplankton size and the species composition affect the pellet sinking rates. So they vary from tens to hundreds of meters per day for a copperpot or a eupausi pellet to more than 100 meters per day for different subspecies. So the fecal pellet flux usually accounts for less than 40% of particulate organic carbon flux, but it varies extremely with the region or the season. So it can be less than 1% or even up to 100% of sediment traps, depending on species, size, and abundance or region. So those can be very highly sporadic events. The active flux is also highly dependent on the migrant biomass and the taxonomic composition. So it can be equivalent to around 11 to 200% of the sinking particulate organic carbon. So these export efficiencies are highly variable and depending on the phytoplankton, the zooplankton, and the bacteria community structure, so to speak, the entire upper ocean ecosystem structure. And it also depends on the degree on the degree of the coupling between primary producers and upper ocean remineralization by the zooplankton and the bacteria, as this is very important also in these highly variable upwelling or coastal upwelling areas such as the Benguela. So what is the motivation behind studying all these components of the biological carbon pump? The global ocean biogeochemical models employed in climate change projections currently use highly simplified representations of pelagic food webs. So to improve these predictive models, there are certain prerequisites. We need to understand the composition, the functioning, and the dynamics at each trophic level with this high seasonal and spatial coverage. So my current research focuses on this mesozooplankton component, currently specifically the copperpods, the amphipods, and the faucids, where we identify the dominant consumers, the trophic interactions, and energetic demands. And my study areas was in the PhD that I did. We took part on a cruise on this meridional transect throughout the Eastern Atlantic Ocean, and I also worked a lot already in the Northern Benguela Current, and now in this current project we have these coastal upwelling areas of the Northern and the Southern Benguela. So our main sampling gear is the multi-net, where we do have different nets attached so that we can study the vertical and the spatial distribution of the zooplankton. We did respiration measurements on board of diverse species as a metabolic proxy, so that we sort the net samples directly on board, choose the key species and make respiration measurements with them on board with this 10-channel fiber optic oxygen meter. And then we can use these respiration measurements as metabolic proxies to estimate carbon budgets of certain zooplankton taxa. So there we can make use of simple metabolic laws, as the respiration rate of zooplankton increases with the increasing body mass, and it also increases with the temperature. So with parameters such as the metabolic rates, individual body mass or length, together with distribution abundances, we can estimate nutritional requirements and feeding impacts of key species and their role in the energy flows. So based on the respiration rates and the community analysis, we can calculate carbon budgets and egestion rates of diverse copperpod, and what I'm currently working on also other communities, to estimate their impact on the vertical carbon flux. So this is a very basic schematic box model here, where the particulate organic carbon is coming into the surface layer as, for example, primary production in the surface layer, and in the deeper layer as sinking particulate organic carbon. This is then ingested by a certain copperpod species or community, which then digests a certain part of this carbon, and this is then, or can then potentially be sink out of this layer into the layer below. A certain part is then lost as inorganic carbon by the respiration, and of course by the secondary production. So this sinking of the fecal pellets and the organic matter, like marine snow, is called this passive flux, which I mentioned earlier. And then we do also have the active carbon flux, where the, in this case, copperpods migrate down to this deeper depth, where they respire the carbon they ingested in the surface layers as inorganic carbon at depth. Yeah, so yeah, why copperpods are specifically interesting to me? Of course, they are often dominant components of the mesozooplankton, and they're also most diverse, with more than 12,000 species described currently. And I chose this picture to show you a little bit this diversity. It's not just their taxonomic diversity, which is fascinating and also important in this cycling of elements. It is also the functional diversity, which means that within the copperpods, they do have very different sizes, very different biochemical composition, or different feeding strategies and energetics. So they do have different roles. They do have different ecological roles. So here on top, you see lipid-rich, larger sized calanoids, Calanus agulensis and Calanoides natalis, which are both key species in the Benguela, and also their young juvenile stages do have these large lipid sacs inside. So they're an energy-rich food source. The abundance is highly variable and connected to this upwelling regime, and they do seasonal vertical migrations. And on the lower, you see small cyclopod copperpods. This is Euthona in the picture, and calanoid copperpods, which is Clausocalanus and Tenocalanus. So they are more energy poorer, but a highly abundant food source in the Benguela, but also in other regions. So they are always there. And on the left, you see a copperpod with a little black dot. This is the genus Pleuromama. It has several medium to larger size species with lipid poorer body content, and they perform diet vertical migration, so they are important for the carbon flux. So different life cycle traits mean different ecological function in the food web and the carbon cycle. So this is from my PhD, where we see that there are differences in the potential impact on primal production of copperpod community ingestion at different latitudes, varying from 21% to 65% of primal production potentially consumed by copperpods. This is the net ingestion, so the egestion is calculated out of this equation, and this was mostly because of different community structures, different abundances, and of course also different primal production regimes, as you can see here. This is to go a bit into detail. This is the vertical structuring here from the epi, the meso, and the bathypelagic zone. This is from the upper figure is from the tropical North Atlantic, and the lower one is from a subtropical South Atlantic region, which was influenced by the Benguela. So it was here in this tropical region, and then here in the southern, this Benguela influenced region, where we did have moderate to high primal production in the surface layer in the tropics and high primal production rates in this Benguela influence region. And we did have differences in the community structure, with the highest surface species diversity in the tropical region and higher amounts of larger carnivores, such as the copperpod Euchyta in the surface layer, while in this Benguela influence region, we just have a high amount of diel vertical migration. So this active carbon flux was higher here in this region. Yeah, I don't want to go into too much detail here. So this is showing the diel vertical migration of this genus I mentioned, Pleuromama. So these are two different species here, where we see that they are doing these diel vertical migrations of several hundred meters within these 24 hours. So they move into the surface layer during the night to feed, and then move back to this into this 300 to 600 meters during the day, where they respire and presumably stay to avoid visual predators during the day. So far, we found around 35 species or stages that undergo these diel vertical migrations at various depths in the northern and the southern Benguela current too. So this is from Schuckert from 2015 in the northern Benguela upwelling system, where we also find different community structures on the shelf and in the oceanic region, where we had copperpods dominating the respiratory flux on the shelf, while further offshore in the oceanic region, the euphausiids dominate the respiratory carbon flux. And even in these deeper layers in the mesopelagic, we do also have the decapods, which are important in actively bringing the carbon down into the ocean's interior. And this is from some work I'm doing at the moment. We also found that amphipods are at times very dominant. This is in the southern Benguela, where the black bars is the total biomass. The blue ones are the copperpods, and these purple ones are the amphipods. And the copperpods were most of the times dominant, but also so there were some events where other taxa were dominant, and the amphipods, which are here in purple, were also very important. And the important thing was also that they were doing diavertical migrations. So this is from a day-night station in the southern Benguela, where we had medium-sized and larger-sized copperpods doing this diavertical migration, and also the amphipod, which is large-sized Vibilia amata, doing several hundreds of meters of this migration. So besides copperpods, euphausiids, and decapods, amphipods are also important in the food web and vertical carbon flux of the southern Benguela. And this, I don't want to go into too much detail of the species. This just to show you this enormous diversity of the colanoid copperpods. This is from the southern Benguela current, where we do have this typical pattern of increasing diversity from the inshore to the offshore region, and also from the surface to the deep. And we identified the dominant consumers along this transect, as these pie charts indicate the proportional ingestion that we calculated for each of these taxa. And so this is just to say that the large functional diversity of the colanoid copperpods is enormous, and it's from small colanoids to large lipid accumulating, seasonally migrating herbivores, from detritivores to carnivores, and diavertically migrating omnivores. And plus, we also saw many different developmental stages, which were also dominant at times and have different feeding strategies in their older developmental stages. So to sum up, the zooplankton community composition has a tremendous impact on the ocean carbon cycle and the vertical carbon fluxes. So we find exponent recycling communities. We have high spatial and seasonal variability, but there are still many uncertainties in the calculations. So the carbon flows through the marine pelagic ecosystems are complicated by taxonomic and functional diversity of the planktonic consumers. And we need to understand their dynamics to resolve this zooplankton black box in the biogeochemical models. So there are recent improvements of these models, which incorporate already plankton functional types, as for example 6 types of phytoplankton and 3 types of zooplankton have been incorporated in rather recent model calculations. So there's a lot of interesting research going on at the moment. And with that, I would like to say thank you for your attention. And yes, I'm open for questions.